Enzymes, remember, are chemicals that aren't consumed in a reaction but can speed up a reaction. One of the major ones we'll talk about this year in AP Bio is called catalase. Catalase is an enzyme. It's found in almost all living cells, especially eukaryotic cells. But what it does is it breaks down hydrogen peroxide. Hydrogen peroxide you probably knew growing up. You'd put it on a cut maybe and it would bubble, or you could use it to bleach your hair. That's pretty dilute hydrogen peroxide. Actually, concentrated hydrogen peroxide, this is somebody who's touched 30% hydrogen peroxide. It damages and kills cells. And so hydrogen peroxide is just produced naturally in chemical reactions, but your cell has to get rid of it before it builds up in appreciable amounts. And it uses catalase to do that. And so if we were to look at the equation, so we've got hydrogen peroxide, or H2O2, is going to break down into two things. One is water, and the other one is O2, oxygen. And so this is not a balanced reaction. So if I put a 2 there, that's 2, and I put a 2 here. So hydrogens, I got 4, 4. Oxygens, I got 4. So perfect. So this is a balanced equation. So you've got 2 hydrogen peroxide breaking down into 2 water molecules and 1 oxygen molecule. But it does that using an enzyme. And so in other words, hydrogen peroxide, let me get my arrow to fit in here, is going to feed into catalase, and it's going to break that down into these 2 products, water and oxygen. And it does that at an incredible rate. I was reading that 40 million hydrogen peroxides will go into a catalase and be broken down into water and oxygen, 40 million every second. And so it's incredibly fast at breaking down that hydrogen peroxide into something that it can use. And so how does it do that? Well, that's what I'm going to talk about. And so basically an enzyme, and let me try and draw an enzyme. So if an enzyme looks like this, it's a giant protein. So if we say it looks like that, it's going to have an area inside it called the active site. And so the active site, let's see how I could do this. Good. So the active site is basically going to be a part of the enzyme where there's a hole in it. So this is this giant protein. It's got an active site, and the substrate is going to fit into it. And so going back to how do enzymes work, well, the active site is going to be an area within the enzyme. So this would be the enzyme here. And basically the substrate fits into it. And so what was the example we were just talking about? The enzyme was catalase. What was the substrate? Substrate is H2O2, or hydrogen peroxide. So that's how enzymes work. It basically tugs on the substrate and breaks it down. It's very important in chemical reactions, and sometimes we want to turn on enzymes and sometimes we want to turn off enzymes. And so in every step of photosynthesis, in every step of the cellular respiration and glycolysis, citric acid cycle, all of those chemical reactions, remember, have to have an enzyme that's associated with them that can speed up that reactions. And so it's really important that we sometimes activate or turn on those enzymes. It's also just as important that sometimes we turn them off. And so there are 2 types of inhibition. Inhibition can either be competitive, that's where a chemical is blocking the active site, or allosteric, when we're actually changing the shape or giving it another shape. Chemical reactions, another important thing that we want to measure with them is the rate of a chemical reaction. And we can do that by either measuring the reactants or the products. So let me stop talking about what I'm going to talk about and actually talk about it. And so here is our enzyme. So our enzyme that we talked about was called catalase. So catalase is going to be a protein. It has a specific shape. And so if we go down here to the enzyme, this would be the enzyme right here, it's going to have an active site. An active site is the area where the substrate can fit in. And so the substrate is going to be this green thing in this picture. It'll fit right in here. And it fits almost like a key fits a lock. And so it's going to be a perfect fit between the 2. Every chemical reaction is going to have a different enzyme that breaks that. And so the important part is right here. So now once we have the enzyme inside the active site, there's going to be a chemical tug. In other words, it's going to pull on that chemical, it's going to lower its activation energy, and so that it can actually break apart into its products. And so if this is our H2O2 right here, there's going to be a tug on those chemicals. Sometimes it'll actually change the pH, sometimes it'll put a mechanical tug on it, but basically what it's going to do is it's going to make it easier for those chemicals to spontaneously break apart. Now hydrogen peroxide by itself, H2O2, if you leave it in a bottle for millions and millions of years, if you come back, it's spontaneously going to break down into water and oxygen. But that's going to take years and years and years to do that. And with an enzyme, it can happen in seconds. Like I said, 40 million hydrogen peroxides can feed through this, create all of this water, and can do that really, really quickly. And so enzymes are ready to go. And so we want to control which enzymes are firing at which time and which ones are. How could we stop that substrate? Again, 40 million of them coming through the active site in catalase. How do we slow it down? Well, there are two types of inhibition. First one is called competitive inhibition. Competitive inhibition is when you use an inhibitor, which is another chemical, and you just get that to bond into the active site. So if you have that bonding in the active site, then that substrate can't fit in, and so we're going to stop the reaction. So if we make an inhibitor that bonds to the active site, we call that competitive inhibition because it's competing for the space with the substrate. Now we can also do that non-competitive inhibition, and we usually call that allosteric. Allosteric reaction works the same way. So here we are, we've got our enzyme. Here's our substrate. It's trying to fit into the active site. We also have what's called an allosteric site, which is going to be another site on the enzyme itself. And so one type of allosteric, or changing the shape inhibition that we can do, is we can have an inhibitor now that's just going to bond to that allosteric site. When it bonds to the allosteric site, it's covering up the active site. And so now there's going to be no way that substrate can fit in. But since it's not actually bonding to the active site, we call that allosteric. Allosteric means different shape, or different shape of the enzyme. So that's a type of non-competitive inhibition. Or we can do it this way. So this would be another type of allosteric inhibition. We can have an inhibitor bond to an allosteric site. But if you look at the active site in this picture, here's the active site. Once this inhibitor bonds with the allosteric site, it now changes the shape of the active site. Once you've changed the shape of the active site, remember the substrate only fits if it's like a lock and a key, and now it's not going to fit anymore. And so this is another type of allosteric inhibition. And so we use feedback loops, and we use inhibitors and cofactors and coenzymes to regulate what enzymes are going off at what time. Now when we do the enzyme lab, we are using catalase. And so when we do it in class, we're using catalase. It's an enzyme. We use an enzyme that's found in yeast. We then fill up a beaker with hydrogen peroxide. We put our little discs of filter paper, or chads, at the bottom. We dip them in varying concentrations of the enzyme, and then we see how long it takes for them to float up. And so what we're varying, or the independent variable, is going to be the amount of the enzyme, and the dependent variable is going to be how long it takes for them to float, or the number of floats per second. And so you could imagine, let me get a better color, if I increase the concentration of the enzyme, we're going to increase the rate of the reaction. But eventually, you can see how it starts to level off here. Eventually, if you have enough of those, let me change to a different color. Eventually, it's going to level off. And so when we're measuring reaction rate, we could measure two things. We could measure the products that are created, or we could measure the amount of reactants that are being consumed. In the enzyme lab, we're measuring the amount of oxygen, so we're measuring the amount of products that are created. But there's other things we can measure in this. Not only the concentration of the enzyme, we can measure the temperature, we can measure the pH, we can measure a lot of different things. And remember, organisms, if we were to measure temperature, for example, the reaction rate's going to increase, and eventually the enzyme is going to denature, and so there's going to be an optimum set point. And since you have internal temperature of 37 degrees Celsius, most of the enzymes inside your body are primed to work at that specific rate. And so that's enzymes, and they are used to maintain that internal balance, and I hope that's helpful.